Hello everyone. You are watching a popular science channel, Breaking Science. And my name is Andrew. Today we will talk about the latest science news. Physics has sections that are not very well known. For example, tribology is a science that studies the contact interaction of solid deformable bodies when they change. This includes friction, wear and crumbling, lubrication, and other processes. Tribology works not only in relation to mechanisms. Recently, with the help of this science, they showed why people love the feeling of chocolate melting in their mouth so much. If a piece of good, dark chocolate gets into your mouth, then you need to know that it turns into a liquid, viscous emulsion from contact with the tongue, chewing and interacting with saliva, and begins to secrete fat, enveloping the oral cavity with a fat film. This creates a feeling of silkiness and smoothness. It is this feeling that is so pleasant to most people, in addition to the sweet taste. We found out this with the help of an artificial tongue. The whole point, as it often happens, is lubrication. It is better when there is more fat in the chocolate, and it is located closer to the outer layers of the chocolate bar. Perhaps this knowledge will lead to the creation of some revolutionary types of chocolate. And yes, if you have a chocolate bar left after the holiday, then you can check out this feeling of smoothness from the melting chocolate emulsion in your mouth right now, while the most interesting science news from the previous week is being released. And as usual, all references to sources and details are in the description. I can't immediately let go of the topic of food, but now it's not about pleasure, but about payback. Scientists believe that they have discovered another harmful property of fast food. In addition to the effect on overweight, cardiovascular diseases, the risk of diabetes, fast food leads to a deviation in the structure of the liver. Specifically, it affects the development of non-alcoholic fatty liver disease. And its development leads to cirrhosis, scarring, and disastrous consequences in the form of dysfunction or cancer. The studies were conducted according to data from 5 years ago. The data of 4,000 Americans were taken, and many parameters were taken into account, including the consumption of fast food. The conclusions were as follows. If fast food occupies at least a fifth of calories in the diet, then people begin to have a moderate accumulation of fat in the liver. If there are still diseases, such as diabetes, or there is obesity, then with the same diet, fatty tissue in the liver will be significantly more than those who either do not eat fast food at all and who have less than 20% of all calories in the diet. In a healthy liver, by the way, fat is no more than 5%. In general, scientists believe that if you even have a snack once a day, it means earning yourself a non-alcoholic fatty liver disease. What associations do you have with the attracting beam? I first remembered the gravity gun and the famous silent, and then the tractor beam from the space simulator. Both devices interact with objects through gravity, but scientists have created an attractive beam that works differently. They used a laser, taking advantage of the fact that light can both transmit a moment, so transmit energy. The solar sail operates on the principle of light pressure in experimental spacecraft, which are accelerated by the pressure of photons. And it's time to remember the optical tweezers, an instrument with which you can hold and move microscopic objects up to the cells, following the laser beam. But, in fact, more can be achieved from the laser by forcing it to move objects along the beam, but against the direction of radiation. In other words, to attract objects. In new experiments, scientists have achieved the effect of attracting macroscopic objects 3 orders of magnitude stronger than the effect of light pressure on the solar sail. But another principle was used for this. The Knudsen effect. The essence was as follows. The objects on which the laser shone were suspended in a rarefied gas and irradiated with a laser, but they were heated from the opposite side from the one on which the beam of light fell. In the experiments, a thin film of graphene and silicon oxide composite on a glass substrate was used. When irradiated, the glass passed a laser, and the composite molecules, receiving energy, were heated from the opposite side of the film, and this led to the appearance of the Knudsen force. When the gas molecules receive energy from the warm side of the film and begin to push the object from its hotter side, so, towards the laser beam. In experiments, this force was almost 1 micronewton. Yes, this material was developed specifically for the experiment. Yes, the size of the attracted object did not exceed 5 millimeters. Although this is already a macroscopic object, not a microscopic bacterium. And yes, this is just a demonstration of the possibilities. But there are still many possibilities for fine-tuning this system, and it is possible that somewhere, in the rarefied atmosphere of Mars, such an attractive ray will find its application. In the essence of the following news, I will inform you that Dr. Reginald Bushrat would definitely approve of the experiment on photosynthesis in mammals. In nature, photosynthesis is inherent in archaea, bacteria, and plants. In laboratories, scientists are experimenting with symbiotic photosynthesis in fish and amphibians, when photosynthetic algae act as symbionts. But there is at least one reason when photosynthesis can be useful for mammals, and that is osteoarthritis. Let's take it in order. Imagine that the cartilage tissue is destroyed due to the fact that some restorative anabolic processes are disrupted in it. Cartilage is difficult to restore because they are not nourished by blood. There are no vessels in them, but they are nourished by synovial fluid. Therefore, clear metabolic processes are important for them. Osteoarthritis, in fact, is the result of a metabolic disorder. Protein synthesis is disrupted in cartilage tissue due to the fact that two substances are missing. Adenosine triphosphate is an energy molecule, and nicotinamide adenine dinucleotide phosphate is a supplier of electrons for the reduction and oxidation reaction. These substances could be delivered directly to the cartilage, and it is inefficient or change the work of metabolism at the biological level, and it is dangerous. Therefore, scientists remembered that plants, during photosynthesis, produce both adenosine triphosphate and nicotinamide adenine dinucleotide phosphate oxidase. This happens inside chloroplasts, the so-called thylakoids, special bubbles. Thylakoids are the very factories that can help with the synthesis of necessary substances directly in the cells of cartilage tissue. To do this, they need to be taken, wrapped in a kind of shell that will hide them from the host's immunity, and run into cells. Scientists did just that. To begin with, they picked up faulty cartilage cells, chondrocytes. Then they took spinach thylakoids and placed them in a shell of membrane cartilage cells, hiding them from immunity. Faulty chondrocytes readily absorbed the resulting vesicle and integrated thylakoids into their intracellular space. Then the cartilage cells were irradiated with light. And this could be done directly through the skin. And then the content of adenosine triphosphate and nicotinamide adenine dinucleotide phosphate oxidase was measured. It turned out that the content of the necessary molecules in cartilage with thylakoids, compared with the control groups, increased significantly, which meant starting photosynthesis in modified cartilage. The experiment, of course, was conducted on mice. It turns out that photosynthesis treatment was successful for them. Cartilage tissue began to recover, and instead of changes in metabolism that are potentially dangerous for the body, scientists took a working tool that was proposed by evolution a long time ago and used it to adjust metabolism and anabolic processes. And it is possible that other diseases associated with metabolic disorders can be treated in this way. I really want to understand, which led to an increase in the human brain millions of years ago. What separated us from the primates and made us who we are now? Many genetic mutations that gradually changed the Homo brain are already known, but now I will tell you about one more. But first, let's remember that there are areas in our DNA that directly encode proteins. These sites are read into matrix RNA, then it enters the cytoplasm of the cell and a protein is collected along it, which then participates in cell construction or other processes. However, not all sections of DNA are needed to create proteins. There are also non-coding regions in large amounts of DNA. They are also transcribed into RNA, but no synthesis occurs. Not all of us know about the role of this RNA, but recently the veil of secrecy has lifted a little. Scientists have studied a long non-coding RNA. It has a peculiarity: it cannot go beyond the cell nucleus into the cytoplasm, but at the same time, matrix RNAs have been found in macaques, surprisingly similar to a long non-coding RNA. After a detailed study, it turned out that mutations occurred in the long non-coding RNA millions of years ago, allowing it to leave the nucleus after all. Maybe then other mutations occurred with her that could lead to a change in the brain. A search through the genes of monkeys and humans showed that they really have genes encoding proteins and sequences similar to long non-coding RNAs, and having signs of mutation that allowed non-coding RNAs to enter the cytoplasm. There were dozens of such genes, but this is a bare theory. Confirming experiments were needed. And then scientists took neuroorganoids, clumps of nerve cells, mini models of the brain, and implanted in them those genes that were active in the human brain. Neuroorganoids, with added genes, grew significantly larger than the control group. But even more interesting is that mice with the same added genes also had a larger brain, better memory, and better cognitive ability. So, non-coding RNA sequences received mutations long ago that allowed them to enter the cytoplasm. There, RNA learned to work with molecular synthesis machines, and then mutations occurred that contributed to an increase in the human brain, and possibly chimpanzees. This all means that now scientists have some tools to increase the brain, at least mice. Well, that's it for today. Thank you so much for watching. I will be very pleased if you like this video and share it with your friends.